Is prepared. I'll go ahead. Got it. I'll go ahead and restart the recording here. Awesome. Well, thank you for everybody for joining us today for developing novel Lyme treatment with Dr. Timothy Hasted. He is a professor of pharmacology and cancer biology at Duke University School of Medicine. His current research is focused on the use of chemical biology approaches to define novel drug targets. These drug targets are focused on the treatment of hypertension, obesity, cancer, inflammatory, and infectious disease, which obviously includes tick-borne diseases. Dr. Hasted received his PhD from the University of Dundee in Scotland, and he's completed postdoctoral work in pharmacology at Howard Hughes Medical Institute, University of Washington in Seattle. In 2022, Project Lyme provided a $68,000 grant through Bay Area Lyme to support his Lyme disease research at Duke in honor of Neil Spector, which we will talk about a little bit more in depth later. But I want to first ask Dr. Hasted a couple of questions about drug development, just so we can introduce the topic for a lot of lay people who may be in the audience. So Dr. Hasted, how do researchers typically receive funding?
For drug development, well, there are a number of avenues you can go to. There's the federal government, obviously, but those programs are kind of rare for raw drug discovery, particularly in the infectious disease space. The NIH is really more orientated at the moment for significant money for drug development would be on the clinical trial side. So a lot of it is private foundations that support what I would call what we're raw drug discovery of new entities that you progress down the discovery path in an academic way. So like we are with the programs that were funded by the Cohen Foundation and originally, and then subsequently Bay Area Lyme. So those sort of foundations, and then you have other ones like the Gates Foundation. They're not really a big Lyme funder right now, and other private foundations here, there, and everywhere, have been a good source. But the process is expensive and to do, but it is possible to do with academia, and it's usually targeted to if you want to do novel things, that's where the foundations really come in to make a difference, like to find something that you could probably then subsequently move into clinical trial through forming a spin out perhaps, or a if you get all the way, like we think we can with the Lyme project, to clinical trial, you might go to NIH perhaps. There's avenues there, or a spin out. Now there is another wonderful program that is funded by the NIH called the small business grants, which SBIRs, STTRs, they can be one way. You could do something like we're doing that Bay Area and Cohen have found, where you're trying to do raw drug discovery. And those grants can be quite competitive for drug discovery, but you tend to have to form a company outside the university, which we have done with something else. But so those are the ways to do it. It's possible for an academic like me to do raw, the sort of things that pharma might do. And where we fit in is in problems like the Lyme problem, which isn't universally necessarily accepted as even being a major issue, by conventional paths. The amount of money that the NIH, for example, would devote to it is very small, comparative to cancer, heart disease, and all the other ones, HIV. But I think we're all accepting that there's a realization even by the CDC that it's an issue, and the groundswell, which should bring more money into the process. I think that covers it. where you
Yeah,
got the, yeah.
I would definitely agree. In terms
Mhm.
of introducing drug development, you've kind of
Mhm.
talked about how you receive funding, what that purpose of it is, which is to, help some of these non, some of the smaller projects, startup projects really get going and develop a proof of concept. And you've also talked a bit about who you've received funding. And the only thing that I don't think you've talked directly about is what type of projects you've received funding for.
Yeah, you're right. Yeah. So right now we're funded for Lyme, to find a novel method of diagnosis and treatment. And our program is actually to develop a PET agent that will not only specifically detect the presence of Borrelia by PET, but also offer a way to treat it as well. So the first goal is the diagnostic side, but our program is actually funded to make something that same molecule, that drug, could be used to not only image the organism but detect it, but also then develop a treatment out of that by killing the organism. And we have made considerable progress on that. We're getting close to what we call a clinical study for or a preclinical study for what's called an IND to move it into patients safely. So we think by the end of this year we should reach that milestone where we start looking for the preclinical development dollars for the IND campaign. So and that work was funded initially by the Cohen Foundation, Alexander and Steve Cohen Foundation, and then subsequently picked up by Bay Area. So we feel pretty good about that. And then the other program we are funded for is by the Cohen Foundation, and it's for the co-infection Bartonella, where there's this idea that the two act together to develop some serious conditions in people. And that is a collaboration also involving NC State with Ed Breitschwitz, who's an expert in Bartonellosis, and Monica Embers at Tulane that has these animal models. Our job, we're funded to discover raw targets, new targets for and we have two or three enzymes that we've been pushing there that they funded us for, that we have interesting molecules that are at the optimization stage. We've tested them in cell-based models of Bartonella or the Breitschwitz lab that had, and we've got a series of 9 of them actually in animal studies right now for pharmacokinetic studies. So that's been very productive work. It's the kind of work that again NIH would never fund. It doesn't really have a mechanism. It wouldn't prioritize something like Bartonellosis or even the combination as even an issue. So it's very important, these foundations that support this kind of raw work. And we don't act in, we have submitted our work for peer review, for exactly how we would operate for NIH, look for high impact journals and put our work through there. So everything is in place to ensure that what we're doing is actually what we say it is. You've sort of frozen. Are you still there? No. I think you have lost the link. I think, yep. I think we're still, I'm not sure we're live. Are you there? I think we've lost our link to our host. What's the Q&A say here? Okay. Yes, you are still live, Timber. I think we've lost our host. I shall try to contact him. It's key that we talk to him because he has the questions. Apologize to everybody. I'm not running this. Let me see if I can reestablish contact. Having a brief pause in things. Of course, I've lost my email. Thank you. Yeah, we're missing our host. Good grief. Nope. Sorry, we don't go away. We'll be back in a minute. I'm going to try to see if I can get hold of the host. Okay. He says, Noah says he's getting back in, guys. So hopefully.
LOL. Yeah, apologies. No idea what happened. We had really not sure, but I know, just to wrap everything up, I know what you were talking about. You mentioned you're working on 2 different projects, one focused on obviously Borrelia and the other being Bartonellosis.
Right.
And you also mentioned molecules. I think that's a good segue into another question about the concept of a small molecule drug.
Right.
Are these 2 projects you're working on considered small molecule drugs? If so, can you explain a bit more about that and how they work?
Yeah. I mean, why don't we have a quick screen share just to give some people some perspective?
Yeah, definitely.
Do you have to hand me, or am I good? I'm there. Okay, I see you. Okay, share. Okay. So hi everyone. I won't give a lecture here. I'll just touch on what a small molecule drug is. So I mean, if you think about the drugs in the cabinet, aspirin, ibuprofen, those types of things, antibiotics, those are all small molecule drugs. They're small in their size. So in this slide here, this is, the structure of aspirin. It's chemically, it's its chemical structure. And this is another cutting edge drugs they use to treat cancer. Since aspirin was, for willow tree many years as a natural product, and this is a human derived drug, been synthesized in a laboratory, and it has something called a weight, which is an atomic weight, and it's for those of you who know what that means, to qualify as a small molecule, it would be anything under what's called 500 Daltons, which is atoms. And it's discriminated from other types of drugs that you might hear about. If you have RA, you might know a drug called Remicade. Or if you've had breast cancer, HER2 positive, a drug called Herceptin, or a vaccine, like an antibody, which is a big molecule. It's a protein of 100 times bigger than these things. And to make those, you need biological systems to make. If you think about the COVID vaccine, that's an mRNA drug that's injected into your bodies and cells, and then your cells make the proteins that produce the vaccine. They make viral proteins. So when we say small molecule, you think about tablets that you eat, put in your mouth. They're convenient for people, when you go on a course of antibiotics like doxycycline for Borrelia infection. If it's detected early, you take one a day for 3 weeks, and that's what we're shooting for to some degree with Bartonella to begin with. But how do they work? Just to bring in what they generally do. You have your drug, this small molecule drug, and it will, this sort of schematic will be a protein. and they bind in something called an active site of the enzyme. And this drug usually mimics some natural substrate in our bodies. And the human, we're trying to make a synthetic version that is better than that natural product in the human body. It'll be commonly, it could be a vitamin binding site. It could be something uses an energy molecule, something called ATP in the body. That's our power engine for, you might compete with that. And what we do as medicinal chemists is make something that basically outcompetes that natural thing, because we've decided that, in the case of an antibiotic, we need to block this enzyme in the bug and not the human one. We're trying to discriminate human so that it doesn't work anymore. And that's how you get a therapeutic outcome. And that's sort of the definition of a small molecule drug. It's something that binds to a protein in a selective way. And with Borrelia and with Bartonella, we're trying to design a drug that recognizes proteins expressed by those two pathogens to kill their activity selectively and not the human form. So that, then we get a positive therapeutic outcome without side effect. And, that's how antibiotics work. That's how aspirin works. And it's how all the sort of drugs that we take orally work. And if you want to look at it for real, these things are real things. So we are able, as biochemists and medicinal chemists, to determine the structure of these proteins that bind these things. And this is what we're doing in the Lyme project and in the Bartonella structure, determine their molecular structure. We use a process called crystallography to do it. And then we can see our drug, how it binds to the active site. This is one that I made that in a company I formed years ago that was acquired by Pfizer, targeting a human protein called HSP90, where we crystallized that protein with the drug that we had made synthetically. And this is a picture of it, and that's how it looks. If you were shrunk down to Ant-Man size or lower than that, you could see these things. That's what they would look like. Proteins look like, with all these folds and topography. And then there's this site here that normally this molecule called ATP would bind to, and our thing beats that thing and has a more affinity by several thousandfold and outcompetes the ATP. And it just, the compound is designed just to fit in this protein and not others. And it's small. It's not that's what we call a small molecule. The protein is a big molecule, and these are small molecules. So, and they compete here. This, and that's pretty much all the things that were the targets that we are working on in Lyme. I think that's all the point I wanted to make about what a small molecule was and what a drug is. So, but the targets we have are behaving similarly. There's in Lyme we have a target called HTPG, Borrelia HTPG. It's a stress protein that protects the organism from heat stress. And then in the Bartonella case, we picked two targets there. We're working on currently. One is called glutamate synthetase, and it's a way to fix that the organism will fix ammonia. We targeted that, and we have a co-crystal structure we just got literally last week for a novel molecule that we discovered that inhibits Bartonella in culture in the Breischwitz lab. So that's really going well. And another one called PRM, which is involved in making actually these ATP type molecules in cells. That's essential for Bartonella. So we got 2 bites, the cherry, if you like, there. And we actually also have a third one on the way. We're just starting, just in case everything fails. So that's our process, is that, we determine the target structure. We find something that binds to it, and we make it synthetically. Our goal is to turn it into a pill. So, and it will, a small molecule pill. So I'll stop sharing for that question, so you don't get a lecture.
No, this is extremely interesting. And I think it's really awesome that you have so many people that you're working on this with across the sphere of Lyme research. Obviously, you're clearly have a track record of success in this, and you're at a major institution. So this is exactly the type of work that I think patients and obviously advocates want being done in the space. So very exciting. I am curious, since you're developing this as a small molecule drug, is there any other existing small molecule drugs that are being used to combat Lyme disease?
Yeah, I mean, the famous one being doxycycline, which is in our hands, it's effective so we're quite an unusual group because we're medicinal chemists. We have a medicinal chemistry group, and I have industry trained chemists, but we also have in the ability to culture the Borrelia in my lab. We brought it in for the reason was to, so we could test and kill the organism by things we make before we pass it on to Monica and even Ed. So we do that, but our control molecule is the gold standard, which is doxycycline, which in our hands is an effective drug for in culture, in the cultured environment, laboratory environment. Very good, potent drug. Would not argue that that's not a good idea. It's a good idea if you have been positively diagnosed, whatever that is, for a Lyme infection, if they caught it in time. I think for a lot of people it's been good. It's worked, and I would agree with that. What we do know is that if we let the organism grow in these cultures beyond their log, so bacteria double. In our hands, I would say at least maybe 2-3 times a day, we see a doubling of growth of these spirochetes that are the Lyme, and find that Doxy is very effective. That good potent inhibitor blocks that. But if you let them grow till they get very dense and they form these biofilms and big hairy monster things, I think I might have a picture. They form these big, they tangle up and they form these big hairy tangles, and then they become Doxy in our hands is quite ineffective. Actually, it's not really good at really dealing with that culture. And it's possible that you could say, well, if that initial diagnosis was missed and they were allowed to progress into this stage where they form these biofilms, they could perfectly easily see why doxycycline might not work there. So I think that's a reasonable thing. Another drug that is also effective is ampicillin in our hands. It's quite effective at both stages in our hands. So those drugs are out there. Each of these antibiotics have their issues, so with Doxy, I would argue that it's probably true that if you're misdiagnosed and or never diagnosed with a Lyme infection and it progresses like happened to Neil, then there's not much that it's too late. So whether ampicillin would work then in that circumstance, I don't think that's been demonstrated. In our hands, it sort of seems to work, but it's not an oral drug. It's something that has to be given injection by IV, and you'd have to give it every day. And it's either given intramuscularly or IV, whereas Doxy's a oral pill, so that makes it easier to ensure you've got drug coverage and, treating the condition. In most people that might work. But one of the big drawbacks with ampicillin though is if for pregnant women, it's the only option. You can't give doxycycline to pregnant women. It's a tetragenic drug. So I only know all this because my wife, when we were having our daughter in the first trimester, got a bullseye. Well, it was a bullseye on her leg. So she went to and being a physician herself, knew what, this had to be treated, but she knew she couldn't take doxycycline. So ampicillin was the only alternative, and that involved intramuscular injections in the backside. She went through that once. And it was so painful. She was, afraid that the stress of it all would have lost the baby. And then, so she, at this point, I knew what she was doing because she actually was, the other alternative was for her to to sit on an IV every day and have this stuff infused in, through a bag. And she was in the middle of residency and so I can't do that. And so she decided to self-administer this stuff to herself. And she sat there in the kitchen with a stop clock and a syringe pushing this into her arm. So this is a pregnant woman in the first round. And I decided she really knows what she's doing because for someone to do that, and know that it's not going to hurt the fetus that's developing, is really must understand and believe in what she's doing, So I had a huge amount of respect for her again, more respect than I had before when I saw that. I was like, but those are the options. So, so why are we doing this? There's two drugs I mentioned that could treat it. And I think mainly it's because, there's no doubt that if it's misdiagnosed, if it's not allowed to progress like any infection, that, and Neil was, convinced me that this was real, was that, you get into this hidden stage where you've got these biofilms and that can't be accessed, and that brings in immunity, and and I think triggers a longer lasting immune response that results in unresolved disease basically. So our hope, so what are we doing about it? And that's where Neil came to me and asked about what could we do to solve this, and we started to come up with this idea about imaging. Could you see an infection? If we want to, because if you look at what the real issue is here with the medical community, it is convincing them that you have an underlying infection that is the problem to your disease. And my wife is actually a radiologist, so I know a lot about that through her. And I'm also somebody who develops molecularly targeted therapies for cancer that we had used imaging in and taken into the clinical trial. So even, we have some idea of the sensitivity of these techniques, particularly those using isotopes. So theoretically, it will be possible to see, metabolically active Borrelia in humans all over the body with these types of agents that we're developing right now. So I think if we get that diagnostic, into people, and demonstrate that, I think it'll remove a huge amount of doubt. That's the number one priority, is that.
I did want to ask, well, first off, I want to thank you for acknowledging biofilms and the chronic nature of the disease. I think without those factors, we likely as an organization wouldn't exist, because that's a majority of our community is people who've been dealing with symptoms for a long time. And that's why these type of drugs and developing them is so important. But I wanted to go back to, you were mentioning imaging and essentially the diagnostic side of developing this drug is you initially were developing for therapeutics, but you seem to be finding some diagnostic insights within that process. Could you go a little bit more into how you're able to increase diagnostics from this therapeutic?
Yeah. That, this is a bit of a, to understand this might be, again, I share my screen. I apologize to people for doing that, but I think it might be easy to show you. And so and this goes back to Neil Spector, right? So he had asked me to join this program, and I really didn't know much about Lyme disease, and I'm definitely not a microbiologist, but we had done some work in cancer, with malaria and dengue fever and things. We had a little bit of experience in infectious disease on that line, developing novel drugs, in those diseases through collaborations with other people at Duke that came to me, like Neil did. We had some major success with some cancer therapeutics with these molecular targeted strategies, and it really comes out of that. And that sort of shows you the proof in the pudding, if you like, that this actually works. It's not a pipe dream. So if I go share my screen again, and I'll try to convey this without, I don't want to lecture people because I'm not a good lecturer. All right, I need to find the share screen again. Here we go. I'm going back to sharing screen again. This was a little bit about why you would go into drug discovery. You don't need to talk about that, and how we do it. I won't bore you with that. So I'd like to just jump to this part here about how we go about this idea of diagnostic. And what we're going to tell you about is we're going to just start to touch on the cancer stuff, just because that went all the way to clinical, and how we can now translate that to infectious diseases like Borrelia, and it's kind of cool. So and I think, you know, there's something I'm going to hopefully get a plug in for. I think this has implications with other infectious diseases. What we are doing with Borrelia in particular, it's sort of been a bit of an epiphany for me. It's a really great model system to attack bacteria in general and come up with new antibiotic strategy that, you know, and I think that our work in Borrelia, some of which is now under peer review, will show that. And then I think that could benefit the Lyme community tremendously too. Anyway, so the small molecules that we are making are a slight variation on what I just described for a small molecule. So I'm going to show it. We call these tethered inhibitors, and what they are is we have a drug that we have designed from scratch that targets that enzyme active site I spoke about. And we figured out we have a structure of this thing at the molecular level. We know how it's bound, and we can then attach things without interrupting that binding to imaging agents through a linking arm. It's literally a string that is a string of carbon molecules, if you like. They're called polyethylene glycol molecules. And then we know how to attach imaging agents. And these imaging agents can be a fluorophore, like a fluorescent thing, or they could be a thing that you could see in a PET machine, a radioisotope. So we have an imaging. They literally look like this. And we, in cancer, we're known for defining a protein called heat shock protein 90 that appears on the surface of tumor cells that are aggressive. And what we were funded heavily for was to develop a molecule that would target that form that appears on the surface of these what's called metastatic cells. And our molecules were based on, first we made the ligand that bound to this protein that's expressed on these tumor cells. And it only happens in these rapidly dividing cells, and we can target it. And then with the tethered thing, so the drug binds, it binds to on the surface, and it brings along this imaging agent, and then you can see it, right? So that's it. And that's the same principle is happening with Borrelia. So there we have, you know, but they won't normally go into the cell unless they're carried in. You know, that's just another nuance. But they don't act like a regular drug that would just simply travel in and out of cells through a process called diffusion. You know, so there's an active process. So that's what makes us able to discriminate them. And to cut 10 years work from that concept to here, this is a picture of how we took this from data from patients. So I won't bore you with all the details of how we got there, but we discovered first there was this surface thing. We discovered we could make a drug that would bind it specifically, and you could inject it into people. You know, went through all the animals and proved it in different tumor models, and end up taking it through a program funded by the Department of Defense, believe it or not, for breast cancer. But although we end up doing prostates for a while, men were, you know, sorry, it's about sexist, but we did recruit men to study. Simply because they were ready to volunteer for it. And we're actually, the breast one is actually following as we speak. But the first peer-reviewed paper we had on this, which came out last year, was in men with prostate cancer, aggressive disease. And this is the structure of the molecule. And here is the drug bit. Here is what the structure of this arm things. And this was a fluorescent imaging agent, and it was used as a histological tool. And what happened was that this was injected into male patients that were selected to undergo prostatectomy. They were going to have their mass removed. That's the sort of trial that we were involved in, where, you know, they were injected with this molecule. It had been FDA approved. We took it through from what's called a GMP, GLP process, takes about a year. We made stuff that went into injectable vials that was safe to give them and managed by a pharmacy, etc. in the trial. And then there were 12 volunteers in the trial, which is sort of the minimum. And it was sort of blinded. And then what happened was that they were injected with this before they went for their tumor resection the day before. And then the tumor was sectioned, and you can see this picture of the tumor mass piece that came out. And then that got sectioned and it got sent to a pathologist who made his diagnosis. And then those slides were subsequently examined by a fluorescent microscope in one of our collaborators' labs, a guy called Dr. Liley, and the lead physician on this was Takuya Asada, was a Japanese postdoc in his lab. And then they made these correlations. And what they were able to say was they correlated the pathologist's analysis independently, made this analysis of the tumor and regions where he thought there was aggressive disease, metastatic disease. And what they were able to conclude in this paper, which I am on in full disclosure, and made that correlation. Our probe really did find these regions of metastases in these tumors. And this was a big deal for us because it's a real piece of validation that the whole idea that you could develop something that would recognize these metastatic regions. Obviously, what we're in the process of doing right now is turning this into a non-invasive procedure by PET. And then what we're going to do is switch these with another molecule that carries an isotope, which is a radioactive, a medical isotope that's non-dangerous. It's not something you find in a nuclear weapon, although it's produced from the reactors that make those things. It's made by the Department of Energy, but it's a way that in a PET machine, a PET CT machine, you could see the tumor without having to cut it out and use pathology. That's what we want to do with that. So I hope that sort of shows you that we can why we do these visualizations. So what this also told us is something about sensitivity and at what degree you could see these things. And we might have been able to make some calculations of what would be the size of the tumor mass you could see. And it's something you wouldn't palpate. You couldn't even palpate it, but you could see it. It would be a few millimeters in size, a few thousand cells. So that's where we are now. That program is going on in breast, and it's being advanced to PET versions. So, what about how are we doing this for Lyme disease? So this is where Neil came in to me, and he said, "Oh, wow, man, I've seen this stuff you were doing, and I heard your talk, and I heard Dr. Liley's presentations, and could you do this for, in infectious disease, particularly Lyme disease?" And, then I learned about what happened to Neil. I'd known Neil through the cancer work that he was doing, and Neil, like me, had put drugs into clinical trials that were novel for cancer. So that's how we knew each other anyway. But he made me aware of Lyme disease, and then I had mentioned that we'd messed about with diseases like malaria, and even, I think we had a program with the Jordanians actually on leishmania, believe it or not. And we've dabbled a little bit, and we've done a little bit in dengue fever, which is a virus. And so we were really quite tuned into the idea that molecularly targeted therapies or strategies we had used for cancer could also work for infectious disease. And the reason we thought it would work, let's look at the infectious disease like a tumor cell, right? How can we discriminate the bacteria from human non-invasively without having to look at it under a microscope? Well, I can look at Borrelia under a microscope. I can even show you what they look like if people are interested. I'm not gonna, they look like this. They're these wiggly things this is a probe we've just developed that just recognizes Borrelia. No human equivalent, and I'll get back to that in a second. But if you take your iPhone and you put it on a microscope and you look at a field of Borrelia, this is my lab, and they look like this, they wiggle around, right? Well, you can't do that to a human being, right? really can't look in their tissues. So, but under the field, you can see it. And this is actually a drug that is being published. We've made, and after 15 minutes of exposure with this drug, if you look at these fields, you see the Borrelia aren't moving anymore. Within 15 minutes, it prevents them moving, and we know why. And then if you look at them 24 hours later, the whole field is clear. This isn't a culture. So this work just got published, but we can look at it. These wiggly bits are the remnants of these blown up Borrelia with our drug. They literally explode. And you're looking at Brownian motion where you see these little bits floating around, but there's nothing left. And we've actually sterilized the culture. So this has been a lot of fun, right? It's a serious disease, but from our perspective, we make things and then we have this idea about how they're going to work. And I'm just going to tell you how that's going to work with discriminating the Borrelia from human. Like you could discriminate it by seeing it visually down a microscope, but you can't do that to a person. We know that if we think about the Borrelia organism as a cancer cell, let's say it's in there, we're going to target just this Borrelia. How can we do that? Well, if we take the Borrelia genome, which has been sequenced ad nauseum, there's something like 130 novel targets that we've defined in Borrelia that we can use our technology for. Of those 130, there's a protein called HTPG, which is called heat-induced protein G. It's got this wonderful name, right? In the prokaryotic world, and that's its genetic identification. This happens to have a structural relationship with the same protein we use for cancer. It's in the human version. It's called HSP90, and the surface one is called eHSP90. And there's a structural relationship that a conservation of that in the protein at the protein level between these two organisms, humans and Borrelia. It goes back hundreds of millions of years, obviously, but it's conserved. It's called conserved. But the amino acid sequences that make up these two proteins, even though they're overall related, the relationship is only about 60% identity. So what we've been able to do with that is understand how the natural substrate called ATP binds to this one and to this one structurally. We isolate those proteins, purify them, express them, and then we developed a screen to take our existing library that we created synthetically to make this make the inhibitor that I talked about for cancer and adapt it for Borrelia. And it took about a year, and we had the structures made, and we worked with a very talented crystallographer at UNC called Matt Redembo. This is a paper we've just got coming out, and we co-crystallized a molecule that we had derived from our screens and did lots of curves and things that us chemists like to do, just showing you a bit of this to impress you, and determine the structure of the human form and the Borrelia form, BB, Borrelia bird dog free HTPG. We know how it binds, and started to turn it into a drug-like molecule, and then found how we could add a tether to it. And we understand all these little things that are showing over here. This is our molecule, and we understand how it binds to here, how the interactions are, where we can change it and not affect its affinity, and turn it into a molecule that we then can, this is a process. It's our method for doing it. We measure things called affinities. We quantify them, and we end up coming out with a compound now, just to sort of cut to the chase, that it will only bind to the parasite or the Borrelia version and not the human protein. This is like something we're super excited about, and nobody's ever done before. No one's ever made a molecule like this that separate the Borrelia HTPG from these counterparts in the human genome, because obviously you don't want to make something that binds to the human form, right? And this was all started out funding with Cohen Foundation, and subsequently pulled it off with Bay Area funding. So kudos to them. And the idea now is to turn this into a pet agent, right? We've done things with it, like here we have our ligand, and it's, you know, only binds this one, not these other two. And because we can change what we call this payload, we developed something called berserkers, which are molecules that when you excite them with light, they emit nature's bleach. That those movies I showed you, there was a proof of concept in culture that you could show how you could deliver this structure, if you like, to Borrelia and kill it. The other thing we can do with it is put on a fluorophore. For this is all for microscopic work and proof of concept and understanding that our drugs are specific for the organism and not the human form. And we can also put things that would carry radioisotopes like iodine or these metals. And that will be our PET version. And this data here is actually something we're developing an AI tool actually that we think you could possibly use this to look for Borrelia in blood. That's something we just started to do based on some literature we saw. But if you mix our fluorescent version of this structure, just to think back here, instead of here's a drug here, same drug, and then switch this to a, instead of killing the thing, but you want to see it in a culture, this is a very dense culture, it beautifully labels the organism. And we've even made we got from a collaborator had sent us a GFP version. This is a green fluorescent protein that lights up the organism without our compound, and we look for co-staining. And this is actually one of these clusters where the biofilm's forming, and we see those really not light up too in culture. So we know these structures get into the organism, which is actually one of the major challenges with any prokaryotic, is getting in through its cell wall, you know, which is a bit of luck on our part that they get into the organism. And I'm thinking that we're starting to look at some co-blood cultures that, given these distinctive spirochete structures, that some AI interrogation of slides and things might actually pick these up in a blood sample. It's something we started to think about just as an adjunct method that other people are interested in. This is some data that we got on, sorry to show you guys curves and things, but it's some basics. I hope to show you guys that what we do is pretty thorough. We try to really thoroughly understand these things, and all we understand is these curves. They have to, the things we make have to do things, and then they have to quantify it before you can progress it into clinical development. We do a lot of that. And basically, one of the things that we made is this light activated thing we call a berserker molecule, and it kills these organisms at all stages of growth. These are sort of growth curves and things that we get excited about when we add these things. And I showed you these movies where, from one of those curves where we look at them down the microscope and they blew the things to bits. And that's why the culture is sterilized. I think that was all I wanted to say. Oh, this is an interesting movie. We actually was so taken aback by the potency and efficacy in the culture when we added these berserker molecules. These are these structures. There's a ligand, and then there would be something on here that we can excite with light, and it emits something called molecular oxygen, which is nature's bleach. It's what immune cells do when they attack a bacteria. They lead these peroxides, or we attached a sort of similar thing that will generate those in response to light, and then it completely obliterates. So how did it do that? So we actually decided to investigate that pretty deeply, and we worked with somebody who works with a very high resolution technique called electron microscopy, and they did a tomography thing. And what we noticed was that after we activated our drug with the light, how did the Borrelia die? And this is the paper that we have now that's in peer review. There's actually also appeared online in Cell Chemical Biology sneak peeks if you want to check it out. But they produced this movie that's kind of cool. I don't know if you noticed those areas. So this is the culture, and they noticed this, what they call a regional electron density. This is a Borrelia, you can see whole structure, and then they notice the drug induced this structure to form inside. We're like completely excited about this. What the hell is that? You know, and it doesn't normally exist in the organism's biology. But the cool bit was it goes right through the middle of the organism. It's perfectly placed in the middle. So there's something biological going on that our drug is induced, but so what is this? It turns out to be the organism's DNA. And our hypothesis is, and what we've argued in our paper, is that our drug, when you activate it inside the organism, the drug gets trafficked through the cell membrane, this cell wall, into the inside, finding this HTPG protein. And then when we hit it with a light, it causes this local generation of this superoxide only inside the Borrelia. So it's not going to affect the human cell. It's all trapped in there. And then it causes nucleoid collapse, which is their chromosome equivalent, to collapse into what we believe, when we measure this, to be essentially double-stranded DNA, a single piece, perhaps multiple strands, because they have the same chromosome they thought is copied 10 or 11 times. That's the current thinking anyway. To collapse into the middle, and it's a terminal event. They don't come out of that. So we're so excited about this that we believe what it's shown, and what our paper is trying to argue, that this could be developed into not only a diagnostic tool using the PET agents, but also a way to clear an infection, perhaps if driven by imaging. But also it's become a novel. The title of the paper is a novel paradigm for antibiotic development, in which you target a bacterial protein. And by doing so, you discriminate it from the host, which is the human, and then you're able to kill it if you can get it to bring one of these things in. So we're hoping that our paper will not be bigger than the Lyme story, but get people excited enough to take it seriously enough to start thinking about this general approach and moving these things into clinical. But anyway, that's the paradigm where we go from this drug-like molecule to the imaging, and how we do it, essentially how we're doing it, and where we are. So,
well, Dr. Hasted, I think that all of those explanations were extremely helpful. I appreciate you bringing the slides, because I think that helps, especially myself as still a layperson, not a scientist, to understand some of the in-depth information that you were sharing. So really appreciate that. Looking online at our Facebook Live, I see a lot of very interesting, exciting work, etc. So people are happy that you're sharing this information. What I want to do now is move into the audience Q&A section. I've received a couple of questions, and I see some people with their hands up in the attendees section. Dennis Sullivan, hey, how you doing? So glad to see you here. I would ask that everybody submit their question through the Q&A section, and I can go ahead and pass it on to Dr. Hasted as a part of the conversation.
Yeah, are you going to curate that so that I don't get randomly lost in? Yeah. Do you want to ask the questions for the host?
Yeah, I will ask them to you. So the first one is from Emily Applegate. I know it's something you had discussed already, but I just wanted to ask it again, of what is the name of the IV drug? I believe it was the one that you're.
Oh, that's ampicillin. Yeah.
How would you spell that? It's
a penicillin based drug. I'm going to embarrass myself. I think it's AMP. Let me get it right. Yeah, it's AMPICILLIN.
All right, Emily, I will type that answer for you. I found it online as well.
Yeah. I didn't know how to spell it, but it's like
no, we're not in a spelling contest here, so no need to use it in a sentence. So okay, cool. That was an easy one. I wanted to knock that off, and then also we have Deborah Tooker. I just wanted to. I know you had mentioned it briefly, but she was asking about looking into the relationship between Borrelia and Bartonella. I know you're doing work on both drugs. I just wanted to reiterate that for her, that you are, I guess, doing that.
Yeah, that's the collaboration with Ed Breichwitz and Monica Embers. Yeah, that co-infection idea. Yeah.
Okay, so one of the other questions we have from an anonymous individual, but they were mentioning the novel malaria drugs that you had created. Could they be adopted for babesia, which is
Yes.
another?
Yes,
Yes.
yeah. That's one of the things in our paper. I'm going to plug it, which is it's on cell chemical biology sneak peeks, and that's basically what they do there is actually public. The stuff is in peer review right now, but people can read it. But that's the where we think a lot of the value from what we're doing with Lyme that has a greater purpose now, in that if we can show that it works in Lyme disease, then there is a massive unmet need for new antibiotics in general, because of many of our drugs like ampicillin, for example, is a good one. Penicillin is a classic. Vancomycin, all these where you've got these nasty resistant strains, and actually what kills us, to this day, we do these, immunosuppression, people that are immunosuppressed are very vulnerable. You get these drug resistant strains that we don't have an antibiotic answer to. And, when we get old and, we succumb, often that's what we pass away with, is that we get a secondary infection. COVID, a lot of those folks died because of, what COVID had done to the lungs, exposing them to pneumonia and things like that. And we don't have good answers for. So I think that if we can demonstrate with Lyme, big news if we can see the darn thing by PET, and we could say the nice thing about isotopes to the number, what's your infection level and all that stuff. That's a big deal. But then you can do something about it. Why not expand it to other diseases like babesia, malaria, leishmania, parasitic diseases, everything. Really, it's a whole career actually that we, that should come out of this. I mean, I think it's, bigger, it's the Lyme could be the flagship for the whole thing. So.
Awesome. I think that's very helpful. I wanted to share real quick with the audience. I see a lot more questions flooding in now. I did want to share that we will likely not be able to get to every single one of the questions given time constraints and also constraints of the organization scope, specifically with this talk, but I'm going to continue to work down and we'll leverage Dr. Haysted's insights as much as we can. And if for whatever reason we don't get to your question, you can feel free to submit it online to info@projectlyme.org. And I'll do my best. You can
also email me. I'll answer them too. I can answer my email. Please do.
Or Dr. Haysted. Yeah, I will keep in touch with him. And if anything is specific to his work, I will make sure to send that out. So just moving on real quick, we have Aurora. She said, what about bacteria living within the RBCs?
Yeah, I mean, again, Bartonella, that's relevant too, because there's you've got three membranes to transgress. And actually, of the three, the human plasma membrane is actually the easier one to get through. It's a single double membrane, single layer, whereas the bacteria have very sophisticated mechanisms to keep things out. And it's just fortunate that we're able to get into that periplasmic space where there's a lot of active transport. So getting through the human or the mammalian cell wall is much easier through simple diffusion than it is to get into the bacteria. We've been very lucky that our molecule target actually is trafficked. So I don't see an issue with getting into red cells, or any cell. So, yeah.
Okay, this one is from Brian Heaney. He said, very exciting work. As I mentioned, definitely not the only person to say that, but he said, can you tell us more about the IND study and when is the acceptance criteria?
Yeah, no, that's to do with the FDA. It's like convincing the FDA first that you have efficacy in an animal model infection, which we're working on with Amir Holzik and Monica Embers on that side. Imaging will just make it is an easier goal than a treatment. So that's why we're doing that first. And I think that's probably more important right now than anything. And what that involves is a process called GMP manufacture. Like there's a highly rigorous process that you go through a CRO, which is a chemical house that you hire to do that. And then you go into safety studies. For an imaging agent, it would be 2 animals, 2 rodents, rats and mice, and then progressed from there into a human volunteer study. So it's pretty, it takes about 6 to 7 months to go through that process, maybe as long as a year to meet with the FDA. And we have done it 3 times with cancer drugs, so we know how to do it. So, but there is a process, and it's a rigorous one, and it's all about safety. And the nice thing about imaging is that they use these microdosing, non-pharmacological. So they are some of the best studies for human studies where you're not going to hurt anyone. You do a dose that's not going to cause any kind of effect like a drug would. You go straight into imaging. That's the beauty of that approach. So we could move very quickly from human studies and probably to our human studies in newly infected Lyme patients. So that's our goal.
I think what you just said about your experience and having gone through that process multiple times with other drugs is likely why Dr. Spector recruited you so heavily to get you involved, because I know something we talk about in the space is there's a lot of great researchers out there, but not necessarily many of them are in Lyme disease or tick-borne disease specifically. So very exciting to see that experience transferred over into this specific subject matter. But I have another question here from Dr. Sue Eisen. She said, thanks for your brilliant analysis. She has not been this excited since she saw Dr. Spector's present on his genomic concept years ago. Will these micro-targeted drugs be able to be used in a patient with 3 or more concurrent tick-borne infections?
Yeah, I mean, that's the point. It's like the target is Borrelia. Our data show that we've even started to look at different bacterial versions of that HTPG protein to see how much we can discriminate those. And interestingly, the differences between certain bacterial species like E. coli and Borrelia are as big as they are between us and those two. It's to do with evolution. And so, yeah, we can exploit the amino acid differences between 2 species to make a specific, we would say that is Borrelia, and it's not E. coli, it's not another related tick-borne disease. That's what we'll be able to say. And I think we can prove it biochemically and in culture for sure. So in a mixed culture, I think. We've certainly proven it between human and the Borrelia, that's for sure. So, but yes, that is exactly what we need. Yeah, good question.
Okay, so this is going to be the last question for the evening, and I think it's going to be a good segue to move into what will happen after this. So this is from Paul, said, can you share and the citations with attendees so we can locate some of the works that you've described?
Yeah, I'll have to do that by email. So there's quite a number.
I figured. So I figured that would be your response. The reason I wanted to ask it is just to share with all attendees, everybody who's here today, as you know, this is being recorded. What follows this is an email tomorrow morning you'll receive from myself with the link to these recordings. It may take a little bit longer. If Dr. Haysted is willing to send those links, I can send all those links at the same time, all in one email. So that's kind of my goal following this is
these will get you these will get you to PubMed and the public database where you can download them all, and everything I've spoken about is in peer review published, and the newer one even which is our latest paper through online, a lot of the cutting edge journals. One we like is Cell Chemical Biology. They'll publish even what's being reviewed, which is a big step forward. So we're positive that confirmation that people are interested enough in what we work is it's to get into the higher end journals, to convince the editor that it's worth, and we made it actually. So we're really excited to see even in review right now. So and you can, like I said, you can access that manuscript online now. So in all its glory.
Awesome. It is very exciting work, Dr. Hasted. Thank you so much for taking the time to speak with me today and share your knowledge, wisdom with all of our attendees here. Thanks again, and looking forward to potentially working with you in the future.
Yeah, thanks for your interest.
Thanks. Have a great rest of your night. Bye-bye.
Bye, everyone.